Do our bodies produce their own light? And if they do, can our retinas actually detect it? If so, what does that light look like, and what can it tell us about ourselves and others? Does this light change when we're sick, and does it stop when we die? In a recently published article, researchers have used extremely sensitive cameras capable of detecting single photons to look for this ethereal glow and investigate its source. Join me as I take a deeper look into the biology of our body's intrinsic glow. I'm Professor Sunny, and thanks for joining me as I start asking questions. For millennia, there have existed reports of people capable of seeing an eerie glow around living things. Often called auras, the supposed glow has been relegated to the paranormal. Is it physically possible that living things produce ultra-weak photon emissions? And if so, are these emissions strong enough and frequent enough to stimulate the human retina? Is it possible that some people could be more prone to seeing this light than others? In the Journal of Physical Chemistry Letters, a cross-discipline team of scientists from quantum physicists to translational biologists have used extremely sensitive digital cameras with high quantum efficiency capable of detecting the number of individual photons per second across a wavelength that includes the visible light spectrum. The researchers placed rodents inside of an enclosed dark box maintained at 37 degrees Celsius and let them acclimate for 30 minutes for the purposes of allowing them to reach thermal equilibrium with their environment. And then the researchers captured a photograph by recording the animals in the box for a total of 60 minutes to attain an average number of photons per second mapped across the animal's body. Before we get into their findings, we need to know what some possible results could be. First, there exists a physical phenomenon called black-body radiation, which refers to the amount of electromagnetic radiation naturally given off by matter which is in thermal equilibrium with its surroundings. In any given amount of thermodynamic energy, electrons are being excited to a higher energy state and relaxing back to their resting energy level. As they return to rest, they emit that extra electromagnetic energy in the form of a photon. Planck's law accurately models this black-body radiation emission spectrum. An object's black-body radiation is dependent only on its temperature, so all matter at 37 degrees Celsius will release an identical black-body radiation spectrum, and that radiation spectrum will be the same across the surface of the object, assuming the temperature is uniform. So this is why the researchers maintain the animals at a constant temperature and let them come to thermal equilibrium by placing them inside the box for 30 minutes before making any recordings. Black-body radiation is, in some respects, the principle behind the light given off by stars and how incandescent bulbs glow. During the 60-minute camera exposure, the researchers recorded an average of around 400,000 photons per second, with variations across the rodent's body. When dead rodents were observed at the same temperature equilibrium of 37 degrees, the average number of photons detected decreased significantly to around 250,000 per second. This means the source of the electromagnetic energy observed was not from black-body radiation. Similarly, freshly cut plant leaves exhibited changes in these ultra-weak photon emissions. So yes, living things emit photons, and those photons are significantly reduced after death. But where's this light coming from? Fortunately, we know that the body's metabolism produces energetic electrons in the form of reactive oxygen species, which are oxygen atoms carrying extra electrons, which are naturally produced during normal metabolic chemical reactions. Sometimes these reactive oxygen species interact with other molecules like lipids and proteins, resulting in oxidation and electron transfer. As these higher energy electrons come to rest in stable covalent bonds, they release their extra electromagnetic energy as photons. And yes, these same ultra-weak photon emissions have been identified from these reactive oxygen species in isolation. So it's our normal metabolic processes that produce this small amount of light given off by our bodies. In fact, basically every chemical bond formation involves this process of electrons being moved to more stable, lower energy states. Chemical reactions are producing photons all around us all the time, but mostly low energy, low wavelength photons. Each chemical reaction would produce a different discrete wavelength of light, so depending on the metabolic profile, the light being emitted might show up as different colors. Now, is this light actually intense enough, or are our eyes sensitive enough to see it? Back in 2016, researchers convincingly concluded that single photons can be detected approximately 23 degrees away from the central fovea of the eye in a region where highly sensitive rod cells are most densely packed. The researchers came to this conclusion after testing volunteers who were exposed to two separate intervals or tests of 1 millisecond in duration, separated by 800 milliseconds. An interval or test may randomly feature a single photon emission. Volunteers were asked to indicate whether an interval in each pair of intervals featured the photon emission and include their confidence on a 1 to 3 scale. The intervals included an auditory signal to alert the volunteers, and the volunteers also received auditory feedback indicating whether their response was accurate or not. The volunteers were given training sessions where they could flip a switch to receive 1 to 15 photons on their retina so they could become accustomed to the stimulus and identify it later. The results showed that the volunteers identified the interval or test with a single photon at a probability slightly but significantly above chance, especially when they reported high confidence of having seen the single photon. Also, it seems single photons sensitize the retina to correctly identifying single photons for a short period of time. This makes sense because retina cells, like most sensory neurons, can become sensitized to stimuli as each stimulus brings them closer to their action potential tipping point. Earlier work showed that individual rod cells have a remarkably high quantum efficiency for a biological system and a minuscule level of noise in complete darkness. But this more recent work suggests we humans are actually cognitively aware on some level of individual photons, at least in total darkness, even if that detection is only slightly above chance levels. Now, it's generally known that rod cells are specialized for low light vision and that they become saturated in brighter light. But how bright? Can these rods still detect light at this low of a level in daylight? In 2017, an international team of researchers tested the sensitivity of retinal rods to produce a neuronal response, and counterintuitively, they found that rod cells have a much faster recovery rate at higher light intensities, at least in their genetically modified rodents. These mice were modified to not produce any cone cells in their retinas, but only to produce rod cells. By doing this, they could test how the brains of the mice reacted to different intensities of light being shown only on rods, without any cones to confound the data. Although one could question whether the lack of cone cells caused a change in the physiology of the retina, the results do suggest that rod cells are active photoreceptors at daylight levels. The caveat here is that this activity was in response to flashes of light across the entire retinal field, not the ability to discriminate image details during daylight levels. While it may be possible for humans to guess at the presence of an individual photon in pure darkness at a rate that's better than chance, and although rod cells are individually highly specialized for quantum detection of photons, I would still be skeptical about the ability for humans to perceive the 400,000 photons per second compared to the 100 quadrillion photons, that's 17 zeros, per square centimeter per second present during daylight conditions. However, the research does suggest that on the individual cellular and brain circuit level, the retina and the neurons of the eye may feasibly be able to distinguish that level of contrast of the intensity of these photons from reactive oxygen species interactions and bond formations. So, are auras real physical phenomena? I've never perceived one myself, but if our hypothesis is that different metabolic patterns create different chemical reactions that emit photons of different wavelengths, then it does seem that we have a testable hypothesis. Until next time, be sure to keep asking questions. Are you a researcher that wants to make your study bulletproof? Do you want somebody to critically evaluate your scientific writing or to develop your latest grant proposal? Maybe you're a journalist and you want to do more than create a click-worthy headline. Or maybe you're a business leader that wants to find out if you can make your processes more efficient. If so, let me know. I have nearly 2 decades of experience across program management, process development, market research, scientific writing, and consulting. And I enjoy applying scientific and diagnostic thinking to hard problems. And if you just enjoy hearing how science works, then please like and subscribe.